All right, so what I want to show you now is how we would present that information that was just calculated through Excel, or whatever other data analysis system you used. So what I have here is my data collection and processing section. I've written it up as it would be expected for a lab submission, and so I want you to pay attention to some formatting issues. So here I've got some labels in a very specific manner. I want you to be able to model that with your own data. This is raw data table 1, the height of fescue seeds measured in centimeters plus or minus 0.05 centimeters. That plus or minus 0.05 centimeters is my uncertainty. And whenever I have a value, if I don't have the unit uncertainty, then it's not complete and you're going to run into problems because you're not fully communicating something. Given different percentages of light exposure measured, well, I've said percentage here, so I could say percentage again at the beginning of this unit, but it's already here. But my uncertainty is plus or minus 0.5% for that value. I transferred my data from Excel into this chart. I've added my qualitative data that I collected during my data collection. And then what I have down here is my process data. So anything that I processed has an additional decimal point, just as I said in the Excel video. So I've got my percentage of light exposure. Pay attention to the titles here. I've got my mean and my standard deviation. My mean has a value and uncertainty. My standard deviation does not. And then what we have here is process data table 2, which is my ANOVA values, and process data table number 3, which is my t-test values. And I could have more charts, or excuse me, more tables if I did further t-tests. If my ANOVA test showed that there was no significant difference, then I wouldn't have a t-test chart at all. And then what I've done is transferred my graph into this and updated it. I've given it a better title that indicates that the error bars are plus or minus 1 standard deviation. I've added my uncertainties, and I've told you that the data represents the mean, not the raw data, but the average. Each blue dot here is the average. I've added uncertainties to my labels. And I would highly recommend that you describe the data. So what I would suggest is that you say that the mean fescue seed height of at 40 and 45% have ranges with plus or minus 1 standard deviation that overlap, which show that there's not likely a significant difference. They also overlap with the value at 65% light exposure. So these three groups are not likely to be different from one another. At least not significantly different from one another. I could pick a value, any random number. If I measured a seed at seed's height, I would not be able to tell you the other way around with how much light exposure it received. If there was a plant that received that was 3.1 centimeters tall, it fits into this data range for 40%, it fits into this data range for 45%, and it fits into this data range for 65%. So these are not significantly different from one another. There seems to be a significant difference between 60 and 65, and if I really wanted to know that, I would have done a t-test. But what I do see from my t-test is that there's a difference between this group at 45 and this group at 50. That would also indicate that there's a difference between 50 and 40, and 50 and 65. But to actually verify that, I would do the hypothesis testing of a t-test between those groups, if need be. And I would describe all of that down here. And I hope that makes sense, and I'm going to stop it there. I'm under 4.5 minutes.